Okay, so as I said, we're gonna continue with prokaryotes and add in eukaryotes to this topic, to this discussion. So I color coded it a little bit, right? So prokaryotic, we've already kind of started this. We're going to continue talking about these structures, their appearance, their composition, their function, and then we're going to compare those to eukaryotes. And for eukaryotes, right, a little bit more complexity with these guys, especially with their cytoskeleton. A major objective for this unit is being able to look at and compare similarities and difference between the two cell types, prokaryotes and eukaryotes. And so remember, whenever we say bacteria, which one should pop into your head? Prokaryote, right? And archaea are the other ones, although they're not as very common. So if they're talking about bacteria, they're talking about a prokaryote. Right, so this is a good organization of information, and we'll fill this in as we go. So we've already kind of looked at prokaryotes last time, so I kind of flipped this guy on his side so we can do a little bit of compare contrast. These are two microscopic eukaryotes, and I'm kind of disappointed in our book they don't have an algal cell, right? So what organelle would an algal cell have that's not represented by these two other cells? Algae. What do they have organelle-wise that allows them to grow? Chloroplasts, right? So you don't see any chloroplasts here, right? This is a yeast, which is a fungal cell, right? This is a protozoa. It could be like euglena, something like that. Notice it has a flagella, just like our prokaryotes. Some of them have flagella. Some eukaryotes will have flagella. But as you'll find out, even though both of them are used for locomotion, they're structurally quite different, and even how they move because of that are quite different. Eukaryotes, by definition, have a membrane-bound nucleus, but within the nucleus they have a region referred to as the nucleolus. That's not an organelle. And they have lots of organelles, and these organelles are organizations of areas, right, that are membrane encased, just like the nucleus is. And actually the nucleus is actually two layers of membrane referred to as an envelope. But your endoplasmic reticulum is mostly specialized sets of membranes. And your cells tend to have two different types of endoplasmic reticulum. One that's referred to as the rough because it has what attached to it? Ribosomes. And then the smooth ER, which doesn't have the ribosomes attached to it. Another set of membranes that almost look like they're stacked coils on top of each other are your Golgi apparatus. And these are important in packaging molecules together. So stuff that comes off the rough endoplasmic reticulum will sometimes go to the Golgi apparatus. So I affectionately refer to that as the packing plant, right? Kind of packages stuff together and sends it off to its individual places. Water vacuole here, again, is membrane encased. In this case, it's holding water. Lysosomes are another vacuole that sometimes you're familiar with that hold digestive enzymes. And are important in some processes. Mitochondria, again, multiple membranes. And those are the energy centers for these cells, unlike algal plants, which have chloroplasts. And they'll have mitochondria too, by the way. But photosynthesis is only your chloroplasts. Only your chloroplasts. What did I forget? Of course, all cells have ribosomes, right? So it's scattered throughout the cytoplasm as well as sometimes attached to the ribosome. They have a membrane. This guy actually has, what do they call this other layer? This guy. Oh, a cell wall. This one has a cell wall. Algal cells, fungal cells. A yeast can sometimes have cell walls. Protozoa do not, right? But this guy tends to have an additional substance, glycocalcics, that's usually associated with the membrane that helps protect it. So when we talk about cytoskeleton, cyto meaning referring to the cell, right? Skeleton within. Just like our skeletons within us help hold and support our cells, the same is true for prokaryotic and eukaryotic cytoskeletons, right? They're going to help that cell maintain its shape, right? So help determine cell shape. They also help when the cells are dividing, right? When you're splitting from one cell into two cells. And this is quite complex when we talk about eukaryotic cells because of that division of the chromosomes that needs to happen, right? All that genetic information needs to be easily divided. And there are special components of the cytoskeleton of eukaryotes that actually achieve that. But stuff doesn't also have to need to be equally pulled apart in prokaryotes as well. It'll help localize proteins in certain areas of a cell. So even though you don't have membrane compartments within a prokaryote, you still have compartmentalization that will happen, and movement within the cell even of things around. And the cytoskeleton helps facilitate those things. So when I was sitting in y'all's seats, I was taught that prokaryotes don't have a cytoskeleton. And in fact, they did. We just didn't have a way of being able to detect it at that time. With the much more high-powered microscopes that we talked about last time, and the molecular techniques and the different staining techniques we have, they were able to, see and even stain, as you can see here, within cells, particular proteins that are attributing to these functionalities of support and of helping move stuff around and helping the cells divide. Right? We just didn't have the ability previously to see this. So now that we know for sure they do have a cytoskeleton. Theirs isn't quite as complex as eukaryotes, though. Eukaryotes, as you see here, have many components that make up their cytoskeleton. We have our really small ones, the microfilaments, the intermediate filaments, and the microtubules. You can see it can create quite a network within the cell, quite a bit of scaffolding to help support that cell and the things within the cell. So the first one that we're going to look at in a little bit more detail is our microfilaments. And these guys are involved in cell motion and shape changes. So especially organisms like amoeba that don't have cell walls, right? And we know they can very much distort their shape. And how it is they distort their shape is that literally these microfilaments are moving, pushing from the inside and pushing that membrane in the cytoplasm. And then movement even within the cell. Pigments sometimes you'll see moving around in a cell. This is because of the microfilaments pushing and pulling them around. So, as I said, in amoeboid ones, that movement is because of these filaments, these microfilaments within the cell. In slime molds, you can really see this movement. Another cool thing that you'll see is that plants, when you put them under the microscope and you shine a light, chloroplasts will actually move within the cell closer to the light. And again, this is made possible by these proteins moving them so that they're oriented in such a way that they're closer to that light source. The next ones are intermediate filaments, and as the name implies, they're intermediate in size. They're a supportive layer within the nuclear lamina, so within the nucleus itself, too, there is scaffolding and structural support for the chromosome, for that DNA and those proteins that are present there. It's also why the nucleus itself, for a lot of eukaryotes, has a specific shape, and it holds that specific shape, and can help us in identifying organisms. Most of the time you're used to seeing this nice, perfect, round nucleus, But if you look at white blood cells, their nucleus gets quite distorted. If you look at other unicellular organisms, one of the ones we look at in lab, Velatinium coli, it has this characteristic curved kidney bean shaped nucleus, right? And the reason why it has this particular shape is because these filaments are there holding that shape. And then when you have multicellular organisms and you're holding cells together, right, to form tissues, these are the components that are helping hold those cells together. So the muscle fibers, right, those fibrous cells that make up our muscles, there's a lot of these intermediate-like filaments holding those cells together so that when they're contracting and relaxing, they'll rip apart from each other. And the last we have are microtubules, and they're actually tubular in nature, so they're hollow in the center. And again, these are really important for cell shape. And when it comes to movement, they work with the microfilaments. And again, intercellularly, they're going to help move things around within the cells. And so a specialized microtubule is referred to as the mitotic spindle. Right? This attaches to the section in the chromosome, that middle section, and to the poles of the cells and helps pull apart those chromosomes from one side to the other when eukaryotic cells are dividing. Cilia and flagella, which actually extend outside of the cells for eukaryotes, are made of microtubules. And cilia are unique to eukaryotes. Only eukaryotes have cilia. But as we've already seen, flagella are present in both prokaryotes and eukaryotes, but they're structurally different. Remember, microtubules, these are a eukaryotic structure only, right? So it's only the eukaryotes, flagella, that have this, not the bacterial flagella or prokaryotic flagella. So the next prokaryotic structure are inclusion bodies. And what they're made up of is either organic or inorganic material. And they're encased in protein and phospholipids and proteins, but again, not organized to a membrane level. We're not talking about organelles here. And so in these pictures, you can see lots of inclusion bodies, right? So here's these areas, right, and the dots here, and then pointing to the arrows here, and all these dots here, right? These are all inclusion bodies. Their functionality, for the most part, is storage, right? So when we have a lot of something, we want to store it up for a rainy day. In doing so, this reduces osmotic pressure, especially for these single-celled organisms. Osmotic pressure can be a real serious problem. So things in solution are what affect osmotic pressure and what we refer to as the movement of water from an area of high concentration to low. Typically there's much more water outside of the cells than there is inside, and so therefore it would tend to rush in and cause those cells to burst. Remember, the cell wall helps prevent that, but anything else that we could do to help stop that rush of water internally is helpful. So one of the ways they do that is that some things that will affect osmotic pressure that they want to store up, they're going to encase it in those proteins or those phospholipid proteins to take it out of solution. So probably something that will make sense to you guys as far as solutions work. You ever see those plastic ice cubes? You know what I'm talking about? They're plastic ice cubes. You put them in the freezer, they freeze. You put them in your drink, right? And they melt, but because it's encased in plastic, it doesn't diffuse into your drink. So it's really great for things you don't want diluted, like, say, orange juice. I'm not a big fan of of warm orange juice, right? But I don't like regular ice in it because then it dilutes it. Make sense? So here, it's like having little plastic ice cubes inside the cell. Whatever's in there doesn't affect what's inside the cell because it's encased. It doesn't affect that solution. It's not going to dilute, right? Or in this case, concentrate that solution. Still with me? Thank goodness for plastic ice cubes. Probably understand that concept. So what structures have a similar function? So what are storage structures inside eukaryotes? And these, in this case, would be membrane-bound. Central vacuoles, right? Lysosomes, right? So that water vacuole, lysosomes, right? And again, the main purpose is storage, but it also will help that cell. And so probably my favorite one is the contractile vacuole for paramecium. So water always keeps rushing into those guys, and that vacuole takes up the excess water, and then it contracts and actually shoots it out. So they look like they're winking at you. It's kind of cool. But again, it's a way of storing that water, and in the case of this one, because it can contract, shooting it back out so that the cell doesn't fill up and burst. Lysosomes, on the other hand, these contain digestive enzymes. And you really want to sequester those. You don't want them running around wherever they want inside your cell and digesting whatever they come up against, right? You want to control what they're digesting. So you're going to keep them encased, right? And you're only going to utilize them and only add stuff to them that you want digested, that you want broken down. It's like acid. It doesn't care, right? Acid will burn through just about anything. So you don't want to you don't want to let it run free. Plasmids are mainly bacterial, right? So mainly a prokaryotic phenomenon, very popular in bacteria. But these structures have also been found in some yeasts, which are eukaryotic. What is a plasmid? It's DNA. But this is different from the chromosome, right? So in this case, in the picture, the bacterial chromosome, this is additional DNA. So some bacteria have additional DNA in this form of what we call a plasmid. They typically tend to be circular, like you see here. They're smaller than the chromosome. And they usually carry information that is advantageous, gives it advantages to the cells. So the plasmids that we don't like very much that bacteria have are what we call resistance plasmids. These tend to be plasmids that contain information on how to be resistant to antibiotics. And the bad news with this that we're going to talk about later is plasmids, these extra pieces of DNA can very easily move from one bacterial cell to another. So they can share this information. And that's what DNA really is information. So it's extra. They don't always have it. They don't have to have it. But for them, for the most part, it's good when they do have this extra information, especially it's something like antibiotic resistance. For us, and we're talking about pathogenic organisms, we really don't want them to have that extra information. The nucleoid is a term I will always use for the region where the chromosome is located. And what chromosome actually is DNA, but also the RNA is there, right, that's being made from the DNA, as well as protein is complexed with it. The function of this area is, of course, to store the genetic information. It's not surrounded by a membrane. It's not an organelle. It's been known by several different names in the literature, and I just mentioned these in case you see something similar to that, but I'll always refer to it just as the nucleoid. But it has been known as the nuclear body. Again, not the nucleus. nuclear body. So similar to chromatin is actually one of the names for chromosome body, or nuclear region. But the area, the region where the chromosome is stored in eukaryotes, we know it to be the nucleus. Right? It's actually membrane encased. This bottom picture is a poor bacteria that died, and this is its DNA shooting out of its cell, becoming part of the environment. So it's just erupting. The cell is rupturing. So as you can see here in this picture that's been specially stained for the chromosome, you can see it's quite condensed, and it tends to be relatively centrally located for the most part. For eukaryotes, in addition to this nucleus, within the nucleus, you will have a region or several regions that are devoted just to the production of ribosomal RNA. Because as you saw, tons of those little dots, right, all throughout the cell. Lots and lots of ribosomes are needed for the functionality of a cell. The main workhorses of your cells are your proteins. Not only structurally, but also functionally. They're most of the enzymes that catalyze the reactions that happen in your cells. In all cells, for that matter. So making ribosomal RNA is very important. And this happens, of course, right at the DNA level, from DNA to the ribosomal RNA, that will then leave the nucleus and go into the cytoplasm, will be utilized to form the ribosome. This is not membrane enclosed, right? So here in this picture, you see this region. This is a cross-section of the nucleus. You see this really dense region here. This is the nucleolus. It's just one in this picture. Nucleoli would be if we had more than one. And again, just lots of ribosomal RNA being produced. The nucleus itself, right, is unique to eukaryotes, defining characteristic, in fact. Stores as the genetic information, acts as the command center for that cell, and it's bound by the nuclear envelope. As I said, it's actually a double membrane layer, two membranes. And then you can see it kind of has holes in it. These are pores, of course, for stuff to be able to travel in and out. Ribosomes. So these aren't organelles, right, but they're very large structures within the cells. They're made of protein and ribosomal RNA. They form these globular structures. There's two main components to them, what we call the large subunit and the small subunit. Of course, the large one is larger in size and weight than the smaller one. They come together to form the whole complex that we refer to as the ribosome. And their functionality, as we've said, is protein synthesis. All cells have to have it. Both prokaryotes and eukaryotes have ribosomes. But they differ in the size of these molecules, the amino acid sequence that make up the proteins, and definitely the RNA sequence, because remember, this is how Carl Woese came up with the three different groups: bacteria, archaea, and eukarya. Because even within prokaryotes, you had a significant difference between the RNA sequence of bacteria and archaea. The system of measurement that's used for these molecules is referred to as the Spinberg system. That's the S that you see next to the numbers. This takes into account not only weight, but how it moves through a fluid matrix. So basically you can think about how aerodynamic the molecule is. So separately, these two molecules, one is larger than the other. You can see one's 30, one's 50. But their shape is quite different too. When you put the two of them together in a standard math that we're used to, right? 30 plus 50 would equal 80, right? But this, again, is not a standard measurement system that only takes into account one variable. It's taking into account two variables. So notice the number is larger. It is 70, right, because we are putting two together. But it's not what we would expect because, again, this is taking into account how it moves. It's like aerodynamicness through a fluid matrix. So the only number I want you guys to remember, because you will see it quite often, is 70s is the size of prokaryotes. Eukaryotes, their overall size of their ribosome complex is 80s. So again, you can tell that in general, eukaryotes are larger, right? Heavier. And so there are two subunits. The large one is 60s and the small one is 40. Those numbers you don't need to memorize, but again, you have a smaller and larger subunit that come together to form the whole complex that we know as the 80s ribosomes. So that's most of the stuff inside cells. So now we're going to travel to external structures. And so for prokaryotes, we're going to look at capsules, although they're not unique to prokaryotes, but very common. We're going to talk about that Fimbriae that we looked at in the previous drawing, Flagella and Pili. You need to be able to diagram, or identify a diagram, and describe the arrangements of flagella. So bacteria don't have just one flagella sometimes. Sometimes they have multiple flagella. How it's structured, how is that flagella structured? And flagella rotation. So for bacteria, it actually rotates like a propeller on a boat. For eukaryotes, we're going to look at the composition and structure and function of cilia and flagella. And remember, cilia are unique to eukaryotes. Prokaryotes do not have cilia. What's the function of flagella? Locomotion, right? Not all prokaryotes have flagella. Not all eukaryotes have flagella. And in fact, for humans, only one cell that women do not possess but men do has a flagella, and that would be sperm. Right? They're the only modal biflagella that we have as eukaryotes. But euglena, right? Trying to think of another unicellular eukaryote that you guys may have seen. Euglena is probably the only one that pops in my head. Oh, good old Giardia's got tons of flagella. That protus. And then bacteria, some of them have it and some do not. I have to think. I don't think any cocci have flagella. And think about that. You know, it's raw. It's round. Most of the ones that have flagella are rod-shaped or the spiral shape that we saw, right? And so it makes sense that it would propel almost like a boat through the water, right? So structurally, when we look at the ultrastructural differences between the flagella in prokaryotes and the flagella in eukaryotes, eukaryotes, as I said, have microtubules associated with their flagella. And it's actually in a particular pattern. There are 2 in the center and 9 pairs on the outside. So it's sometimes referred to as the 9-to-2 arrangement. Prokaryotes, theirs is made of a single protein that's stacked up on itself almost like a coil called flagellin. That's real inventive, right? Their movement is different. As I said, this basal body is going to spin, okay, in the membrane. And notice that this organism has a plasma membrane, peptidoglycan layer, and an outer membrane. So is it gram-positive or gram-negative? Gram-negative, because gram-negative are the ones that have what structure did I mention? Yeah, outer membrane, 2 membranes. Yeah. And there's another portion, this is just showing you the building of it. So the proteins are made inside, they go through, and then they stack up when they're making their flagella. But there's actually a hook portion not shown in this picture. And so again, it spins around like a propeller on a boat. And we'll take a look at that in an animation. Eukaryotes, on the other hand, so prokaryotes, P for prokaryotes, they rotate. And they rotate to what's called proton motive force. This is pumping protons across the membrane. That's how they generate the energy here to spin this base. Eukaryotes, their flagella move in waves. So they wiggle kind of back and forth. What similar structure to flagella do eukaryotes have that prokaryotes do not have? Cilia. And the difference is the cilia are shorter than flagella. Flagella are really long, cilia is shorter, and usually a cell that has cilia, they have multiple cilia, much more than they would have flagella. Because again, they're shorter, so you need more of them to create the locomotion. So here's a gorgeous paramecium. Isn't he just handsome? Completely covered in cilia, right? All these hair-like things that you're seeing, that's the cilia. And they would beat, right, or wave to allow it to move. And there's really awesome YouTube videos out there. You can see this stuff moving. Cool. We have ciliated cells in our body too, in our respiratory tract. The cells themselves don't move though. What are they moving that other cells nearby produce? Mucus. Mucus. Right? So your respiratory tract is protected by mucus, traps pathogens. Your ciliated cells beat it up so that you swallow it, and it gets killed by the stomach acids. Right? So this is constantly happening, right? You notice it more so when you're sick because you'll produce even more mucus, try and help trap them and move them to where they could hopefully not create problems for you. Right? Still same thing, movement. Except not the cell, but the surroundings in that case for us. This is showing you really close up of different types of flagella that are out there. So this one is smooth. This one has additional projections. So which one do you think would be more beneficial as you're trying to move, especially through water? Do you want the skinny, narrow one, or do you want the one with the additional projections? You want the skinny, narrow one? You're going to go with the hairy one? Okay, so let me phrase it in terms that you may understand. Do you want the skinny paddle for the boat or the big paddle? We want the big paddle. Right, so this is basically a big paddle. It's a little paddle. That's the benefit of it. Right? Why some organisms have that additional protrusions to the flagella. So again, notice eukaryotes, that wave, right? So this would be cilia, this would be flagella, weaving back and forth. And then for prokaryotes, as I said, they could have multiple flagella. And these are probably electron micrographs, I think, these top two pictures. These two, I'm pretty sure they're probably stained, probably using the flagella stain. And what's unique about that flagella stain? Why is it allowing us with a light microscope maybe to see these flagella? What does it do to that structure? the flagella stain. Makes them thicker, right? Because remember, these are teeny tiny, and we need it made thicker to actually visualize it under the light microscope. So the thickness has been increased, probably for these two. Now there's some terminology that we use for how many flagella they have and where they're located. So this guy, just like E. coli, is completely covered. You see all the flagella all the way around it. And notice again, what shape is this organism? It's a rod shape, right, or a bacillus, okay? The terminology we use for this is peri, meaning many, trichus, which actually means hair-like. So if someone says that one has flagella, peritrichous arrangement of flagella, you know it's completely covered in flagella. If we say mono, we mean one. So monotrichous means one. Also, sometimes you'll see the term polar, right, as in one pole, because notice it's only at one end of this, again, what shape? Rod or bacillus. This guy is rod-shaped with curves, so this term would be spirillum, not the slinky one. The slinky one under the light microscope looks like a zigzag line. Yeah, this is spirillum. But good guess. All right, and then notice it has flagella at both ends, right? But multiple flagella, right? The term for this is lopho, and this refers to a clustering. And this term is used whether they have it at both ends, a clustering, or at just one end. So although this picture is showing both ends, lopho is referring to a clustering at an end, whether it be one end or both ends. Amphi, anyone know what amphi usually stands for? Opposite. Opposite. No one took medical term? I don't know if that's one of them, though. It's a general term, though. But so in this case, it's one at both ends, right? So there's one at both ends. And again, this one is what shape? Spirillum. And there's three of them, so it'd be Spirillii. The Pili, I always want to put one more P in there, Pili. There's two types. Ones that we typically refer to as Fimbriae. And Fimbriae is when they have a lot of them, usually, on the outside of the cell. So notice all these little ones are probably Fimbriae. Notice it says other Pili. Same thing here. Little tiny ones you can kind of see on this picture. These are used, we found out recently, they can be used for movement to kind of twitch along, almost like having like little legs. Kind of crazy. But their main evolutionary reasoning for some of these cells having it is typically attachment, especially when you're talking about pathogenic organisms. Right? So, this is Cilia gonorrhea that causes gonorrhea, right? That particular bacteria has Fimbriae that's specifically attached to our epithelial cells, right? So whether it be in the vagina region or it can actually even cause a form of conjunctivitis. The bad news with this is that those Fimbriae, that protein structure, the organization of it, it's always constantly changing. If it wasn't always changing, then we can maybe make a vaccine against it or an antibody, you know, against it, and we could vaccinate people against it. That would be great, right? You just can't keep up with the amount of change that this guy does. Also, you can get gonorrhea more than once, right? So it explains why you don't develop immunity to it, right? Because it's always changing, especially on the outside. But we all know how you get that, right? that gonorrhea, the clap, they call it, right? You can avoid this. Okay. The good news, too, is that if you do happen to get it, what could you take to kill bacteria? Antibiotics. What's the bad news nowadays with some antibiotics and some bacteria? Antibiotic resistance. Yeah. Just don't get it, right? You got it covered. Whew. It's a scary world out there. All right. So then the next one, oh boy, we're getting to this next topic, right? SEX, baby. Right? Well, that's what this is. In truth, it's an exchange of genetic information. So even bacteria, even prokaryotes do this, right? And they got to join up too to do it. And how they do that is through this protein appendage, right? That's specialized pili that we typically refer to as sex papillae, or fertility papillae, or F papillae, standing for fertility. So literally, two different bacteria can join up by this protein appendage. That's what they're doing right here. Right? This is a bridge between two bacteria. They're getting it on. What's happening? DNA is traveling from one bacteria to another. Same thing for people, right? Little spermy guys, just mobile DNA, y'all. Traveling from one person to another. This is DNA, and in this case, it's extra DNA we call a plasmid. Plasmids, as I said, very commonly shared among bacteria. Extra information. Here you go, friend. You stick one E. coli in a test tube with all his friends who do not have a plasmid, he will give it to all of them. It's just as bad as a sexually transmitted disease. You're going to give it to everybody who ain't got it. Right? You're going to share. Because once one gets it, then they can pass it to someone else, right? So, I mean, that one little guy doesn't have to pass it to everybody in the tube. Once he passes it to one person, they're going to go and pass it to the next, and they're going to pass it to the next, and then it just expands, right? Exponentially. Still with me? All right. Moving on. Capsules and slime layers. So, if they have it, not all of them have these things, right? But if they do, it's going to be the outermost covering. So if we're talking about a gram-negative bacteria, you got the membrane, maybe a little bit of peptidoglycan in that outer membrane, then the capsule, right? Gram-positive, you got that membrane, thick layer, peptidoglycan, and then the capsule. And then guess what? This is a yeast. So, do eukaryotes have capsules? Yeah, because a yeast is a eukaryote, isn't it? Yeah, they do. Some of them do. But, oops, I guess I didn't have an answer there. That one's pretty easy. The answer is yes. And bacteria, remember, typically prokaryotes. And so here's a bacteria here. And so this clear layer you see is the capsule. And we do a special stain for this. We stain the cell and the background, and the unstained capsule, therefore, is seen. Here, it's two yeast cells. You see, this is the cell, and this is the cell. You can see the membrane. Again, you see the area where the capsule is, and then this is the background. It's a little bit different technique for this particular picture and slide. What does this help them do? It aids in protection, right? If you have an extra layer, you're going to be better protected. And then good old attachment again, especially with our pathogenic bacteria. So stuff like Klebsiella pneumoniae, why it causes such bad pneumonia is that capsule helps it stick to our cells, to our epithelial cells within our lungs when you inhale it. It also helps add an additional layer of protection because it's like they're wearing a disguise, right? This extra layer, our phagocytic white blood cells have a hard time recognizing them, grabbing onto them, and destroying them. So that's capsules. Slime layers are similar in that they can help with protection, they can help with attachment, Additionally, they can help the organism be motile because they could use the slime to glide. Structurally, though, they're quite different too. Capsules, as you can see, are pretty uniform. In these pictures, we're looking at capsules, right? Pretty uniform structural layer around the outside of the cell. Slime, when you think about slime, you think of oozy, sticky, kind of movable, right? The capsule is pretty rigid, uniform. Slime, on the other hand, not uniform, irregular, and movable. So this is our table filled in. So prokaryotic skeleton, very long, thin filaments made of protein, used for cell division, move stuff around inside the cell, help determine cell shape. Very similar to eukaryotes in functionality, but for our eukaryotes, remember we have the microfilaments, which is the smallest, then the intermediate filaments, and then the microtubules. And some of those have additional abilities. For inclusion bodies, remember this is a prokaryotic appearance. There'll be areas that are within the cytoplasm, right, that are enclosed, and usually are going to contain a large amount of some similar material. Their shape can be circular or oblong, and the stuff that's in it can be of organic or inorganic origin, right? So it can be something made by the organism or something inorganic from the environment. If they're enclosed, they'll be enclosed in protein or phospholipids and proteins, but again, not a membrane. Why do they do this? Storage and to help with osmotic pressure. Remember the plastic ice cubes. Sequestering. Plasmids, not found in all prokaryotes, right? And it's found in some eukaryotes. It's very common in prokaryotes. What is it? It's circular or linear. Most commonly it's circular, as we saw drawn. It's extra genetic information. What is it? What is genetic information? DNA. And again, it's extra, don't have to have it, but if they do, it's usually an advantage. Nucleoid is, for prokaryotes, irregularly shaped, usually been present in the cell, and it's where the chromosome is stored, right? The DNA, the RNA, the protein. Ribosomes, all prokaryotes and eukaryotes have these, right? They're all made of ribosomal RNA and protein. In drawings, they're going to look like little dots, either in the cell or even on the membrane. They function in protein synthesis. What's the differences? What's the difference between a prokaryotic and a eukaryotic ribosome? Size. Are the proteins the same? Nope, they're different. Is the ribosomal RNA sequence the same? No, they're different. Right. So their composition, right, exactly their protein sequence, their RNA sequence, their ribosomal RNA is different, and their sizes are different. Who's larger? Eukaryotes. What's their size? 80. And what are prokaryotes? 70. We've mentioned these endospore guys before, right? For bacteria, they fall under the grouping of prokaryotes. And again, only some of them have this ability. Not all bacteria can form endospores. Thank God. It'd be a really scary world. So, speaking of scary world, what disease did we learn about that produces endospores? Anthrax. Anthrax. What causes tetanus as well produces endospores. And that's why, you know, we have that problem, is that we can't really eliminate these from the environment, especially tetanus, Clostridium tetani. So what happens? These organisms, right, have the ability to go through this process of forming a spore. This is referred to as sporulization. And they're going to do this when the environment is harsh, when they're not going to be, when they sense that they're probably not going to be able to survive much longer. So metabolically, they're going to shut down, and they're going to surround and protect the most precious thing to that cell. And what is that molecule? DNA, right? Because that dictates everything for the cell. So it stabilizes it, encases it in a protein coat. Actually contains stuff like keratin, right? That's that hard stuff in our nails and our skin. I have that in there in this coat they use to protect their DNA. Eventually, the cell proper, the mother cell, or sporangium, is going to deteriorate, and you'll just have that spore all by itself. But that's why they're called endospores, because they form within the cell. Germination is when conditions become good. So unfortunately, those people that inhaled the anthrax spores, right? That powder was millions upon millions of spores. Those spores can sense that they're in an environment now that is nutrient rich. And they're going to germinate. They're going to reform that original cell they once were. Remember, these terms sound like reproduction, right? Sporulization, germination. It is not a form of reproduction. It's a dormant cell type, right? They're just going into dormancy. There are two genera that contain lots of species that produce endospores. Bacillus, right, like our Bacillus anthracis, and Clostridium, and I have it written on a slide later, I should. Clostridium is a genus that tetanus is in, tetani. So, there they are. And then there's actually a cocci genus that has spore formers, and of course the name kind of tells you, sporo, right? Sarcinia, so again they're forming packets, or cubes of cocci shaped. Not a form of reproduction. When are they going to sporulate? When are they going to form spores? Bad conditions, when conditions are harsh, right, where life is not going to be possible. This can be any number of things depending on that organism. It could be a change in atmospheric gases. Could be oxygen is not available or oxygen is available, or carbon dioxide is or isn't. Could be there's no water, right? All living organisms need water. A lack of food would definitely cause you to sporulate. Temperature changes for some of these organisms. Differences in light. I mean, it just, you know, depends on how crucial that environmental constraint is to that organism and whether it would prompt it to sporulate. So the organisms that we grow in the microbiology lab, for our students to do the spore stain, we starve them. Right? We put them in culture, and there's a set amount of food in that culture. So the longer they're in there, the less and less food they're going to have as they continue to grow. And so then some of them start to form spores. So our students are able to stain and see the spores under the microscope. So we starve them. We're evil. Right. So as you can see here, the cell will actually go through the process of cellular division where it copies its DNA, but it's actually not going to divide into two cells. It's going to take a portion of that DNA and start surrounding it in those layers of the spore coat and protecting it, as I said, within the cell. The other part of the DNA will be used for the cell to continue to function until functionality can't happen anymore, right? And then it'll completely deteriorate, and you have the spore all by itself, what we call the free spore. That, when conditions become good, right, germination can happen. Water has become available again. Nutrients have become available. Oxygen, carbon dioxide. Whatever it is that was lacking that it needed, it will form back the original cell it once was. And then it can start dividing as well, right, and reproducing that way through binary fission. So because these spores form within the cell, and notice again, most of the spore formers are rod-shaped. So if it's a rod-shaped bacteria, bacillus, and the spore forms right smack in the middle, we say it's centrally located. And so this would help us in identifying an unknown organism, because a particular species will always form its endospore in a specific location within their cells. Terminal means it's right at the end of the cell, right on that end. Subterminal, as the name implies, it's not all the way at the end. It's not right smack in the middle. It's sub, right? Sometimes when the spore is formed, it will actually be larger than the cell itself. And so they refer to this as a swollen sporangium. Sporangium is the term of a cell that has formed a spore within. Again, this is in other organisms used as a reproductive term, not reproduction. The spores themselves can be this oblong, oval shape, or they can be more of a spherical shape. This one isn't quite spherical, but pretty close. So that's another thing they'll look at, its actual shape of the spore. So we've talked about one type of locomotion for bacteria. And we've kind of mentioned a couple others, so let's see how well you guys are paying attention. So the most obvious is the use of what for locomotion for bacteria or prokaryotes? Flagella, right? And that rotation of the flagella allowing them to move. What other substance can they produce that would help them move? No, Cilia is eukaryotes. A sub-slime, right? Help them glide. And then what other projection could help them kind of twitch along? The Fimbriae. Okay? And then one of them, their structure, right, is what also helps them move in a corkscrew fashion. What ones move in that corkscrew fashion? What shape of bacteria? The spirochetes. Good. So we have the flagella that moves like a propeller, right? We have the spirochetes that have that corkscrew movement due to the axil filaments that they have. Gliding motility due to slime production. And the twitching via the Fimbriae. These are all possibilities for movement for bacteria. So how do they use this locomotive ability? Well, especially when they're in aqueous type solution, they're going to quote-unquote swim towards chemical attractants and away from repellents. Well, an attractant would be something like food, right? And a repellent would be some type of waste or something harmful, something toxic. But how do they know? They don't have eyes, ears, noses, do they? What do they have? So they're a cell. At the very least, they have a membrane. What's that membrane made out of? Phospholipids and proteins. And those proteins can help stuff move in and out of the cell, but guess what else they can do? They can be receptors and sensors. These are their eyes, ears, smell, touch for their environment. Right? So the protein receptors in their membranes help them sense their environment and are going to help determine which way they're going. Am I going towards that? Does that smell good? Is that food? Or is that bad news? Am I heading the other way? So for bacteria, right, they have a single flagella. It's going to spin in a particular direction, just like a propeller I said on a boat. It's going to spin. So just like a propeller on your boat, when it's turning a particular way, and it's either clockwise or counterclockwise, and I'm not going to have us remember that, right, it's going to go forward. And in this case, we call this a forward run. When they go in the other direction, it causes them to tumble about. They're not very good at backing up, you could say, right? Unlike a propeller on a boat. Which, I don't know about you, backing up still is difficult for me. Which way you turn the wheel, right? They don't have a wheel to turn, right? So they kind of flop around until they get kind of oriented, and then they'll run more straight. Now notice ones that are completely covered in flagella. When they run, they can actually orient them so that they're going in a straight direction. The tumbling is quite tumbly, right? Because then they're all spread out and they're tumbling around. The spirochetes, as I said, have those axial filaments. You can really see the lines drawn here. And again, this is going to create this helical shape, which is also going to allow them to corkscrew through their environment. So I think we're almost done, huh? Is our last ones? Yeah, it is. Last major set. So the last is for our eukaryotes, looking at the composition and structure and functionality of the internal organelles of the endoplasmic reticulum, Golgi apparatus, lysosomes, the nucleus, the nucleolus, which remember isn't an organelle, ribosomes, which are not organelles, mitochondria, which are, and chloroplasts, and defining endosymbiotic theory. So, endosymbiotic theory I mentioned earlier, this is that belief that certain organelles like the chloroplast and the mitochondria were once free-living organisms, probably of prokaryotic origin, due to the fact that our mitochondria and chloroplasts have ribosomes that are reminiscent to prokaryotes, as well as the DNA that these have. They were once engulfed then by a primitive other prokaryote, and they began this symbiotic relationship, right? So that's the symbiotic portion, endo meaning within. And so much that evolutionarily they evolved together, and your mitochondria cannot exist on their own without your cell, and vice versa. Your cell cannot exist alone without the mitochondria. There are certain aspects of your mitochondria that are dictated now by information that's in the nucleus. So that mitochondria can't live on its own. And then, of course, our cells themselves, their major energy powerhouse, that energy that's produced, it's done in the mitochondria, in those structures. So we can't survive without them. And one interesting tidbit is your mitochondria come from your mom. Your mitochondria are in the egg, and the sperm, as I said, is pretty much just mobile DNA. It has a few mitochondria, but those are just really to motor that flagella to get it where it's going. They don't usually contribute to the zygote, to the beginning cell. So the bad news with that is that you're only getting mitochondria from one source, from your mom. And so some genetic diseases that are associated with the mitochondria are inherited what we call maternally from the mother. Dad can't save your butt in that one because he doesn't really give you mitochondria. So like all the ribosomes and like most of your cell, your very first cell, is from mom. And that's why that's the largest cell in a woman's body, because it has all the nutrients and everything for that, the beginning of a new organism. An ostrich egg is actually a single cell. That's the largest cell on Earth, because it's the largest egg. And again, it's all that food inside for that developing. And in that case, I mean, all the development happens in there, no outside. Does that make sense to you guys? Right? So this endosymbiotic, right? That's where these came from. The two organisms, came together and then evolved to the point where they can no longer be separated. And we have distinctive evidence for mitochondria and chloroplasts being their evolutionary beginnings. So as I've said, our mitochondria are powerhouses, right? And they themselves, as you see, have their own DNA and RNA, and they've got multiple membranes within, for that electron transport chain, right? For producing that chemical energy. Photosynthesis, organisms that have this ability do it with, if they're eukaryotic, within the chloroplast. And again, multiple stacked membranes where the electron transport chain is, so we can generate that energy. The starting point here is light energy to chemical energy. It's the only difference. So when we look at endocytotic pathways, these are unique to eukaryotes. One of the major reasons why this cannot happen for eukaryotes, for prokaryotes, is they have a cell wall getting in the way, right? Cell wall is rigid structure, it's not malleable. Cell membranes are malleable, right? And so the membrane can be manipulated and actually brought within, the endo meaning within. And there are certain specialized endosynthotic pathways. Phagocytosis, certain cells have this ability in our bodies where they actually will engulf foreign matter like bacteria and viruses and bring it in to digest within our cells. Endocytosis, again, this is how most unicellular protozoa eat. They actually engulf into a vacuole and bring it within. And even some of our cells do this. We'll actually take stuff in within a vacuole, large molecules. Auto means what? Self. And so this is when we're digesting stuff within ourselves, right? When we're breaking down our own cellular components. And in order to do that, when we're breaking stuff down, we need the vesicles, those specialized vesicles referred to as lysosomes. So in biosynthetic secretory mechanisms and in endocytotic pathways, there are several organelles that are involved in this process. So your rough and smooth ERs, they're going to be responsible for transporting materials, producing proteins and the lipids. They're going to make up some of these vesicles. And then, as I said, they get sent to the packing plant. And what's the packing plant? The Golgi apparatus. And so off of the Golgi apparatus, after they've packaged this stuff up, you'll get the lysosomes forming. And what lysosomes are membrane-bound acidic sites of intracellular, within that vacuole of molecular digestion. So for phagocytosis, as I said, some of them can take their membrane and engulf a bacteria into a vesicle that we now call a phagosome. That's going to then fuse with the lysosome, right, that was made by our cell, and digest this stuff. Sometimes we can take large stuff in from outside, right, that's endocytosis, and then we'll fuse that too with the lysosome to digest what we've engulfed. Other times we will form vesicles around internal components of our cells, like mitochondria that are deteriorated, and then we're going to fuse that within the cell with a lysosome and digest and recycle those components of that mitochondria. So this is just a bigger picture, which I probably should have switched to earlier. But hopefully you guys seen it, right? So phagocytosis is where we're engulfing things like bacteria, right? Endocytosis, we're just taking large molecules in. Autophagy is where we're actually engulfing stuff that's already within the cell into a phagosome to be digested by a lysosome. This table in your book is a pretty good summary table, doing the comparison between prokaryotes and eukaryotes, and even having the subdivision within prokaryotes of eukaryotes. There's a couple components that we didn't go into detail about, so I put a red line through them. So remember, a true membrane-bound nucleus is a defining characteristic of eukaryotes, right, within the domain eukarya. All of them are eukaryotes. Nucleolus, remember that's a specialized region within the nucleus, so again, only eukaryotes have that. Mitochondria and chloroplasts are organelles, right? Only eukaryotes have them. And then remember the plasma membranes, you have a distinctive difference between archaea and bacteria in the bonding, the ester versus the ether. And then look at this. You know, we have the same type of bonding with eukaryotes. It's really just the archaea that are different, right? And having the different bonding. And those monolayers that they can form in the branched. Flagella. Very submicropic in size. One protein fiber, right? That flagellin. And these guys move by rotating. And submicroscopic, they mean like with a normal standard light microscope, you wouldn't see them. What do we do so that we can see them? We stain them, and that stain, remember, increases the thickness so that we can visualize it. Remember I mentioned about the eukaryotes have the 9-2 pattern of those microtubules. And again, how do these guys move? They move in waves, right? They move in waves. Endoplasmic reticulum, Golgi apparatus are both organelles, so only found in eukaryotes. Peptidoglycan in the cell wall is a defining characteristic of, more specific, bacteria. Bacteria. Because gram-negatives can have peptidoglycan too, just usually a very thin layer. Okay, so remember, within prokaryotes, a defining characteristic of bacteria. Ribosomes, size 70S for prokaryotes, 80S for eukaryotes. Lysosomes, again, this is an organelle, right? So eukaryotes only. Cytoskeleton, remember, not very complex in our prokaryotes, but it does exist. Much more complex, right? Many components for eukaryotes. A gas vesicle is actually an example of an occlusion body. And remember, who did we say had these? Prokaryotes, so both bacteria and archaea. All right. I know you guys are packing up, but give me a few minutes. You'll notice that in my presentation I had several places where it says watch animations. And they are associated with your textbook. But what I've found in the past is easier for my students is I usually try to find them in other places on the internet like YouTube. And so I'm trying to remember if I have those links in here. Yep, here they are. So, yep, see, they're all YouTube links. So I'm going to transfer these over like I did for my lectures, and I've been recording this lecture too. So we actually have the visual now. And I'm going to go ahead and stop that.